scientist, I'm a biologist, and yet I have been part of, I cannot call it an astronomical event, but last year I was part of a little research project about bird migrations. I hope आपको सबको idea है bird migrations क्या होता है. Constantly across the sky, birds are migrating. And there are winter migrations and autumn migrations, and we were wanting to record the migrations of birds. But these migrating birds are so high up in the sky, आप उनको देख नहीं सकते. And the migrations are such कि one of you had asked कि simple equipment से कैसे हम कुछ कर सकते हैं astronomical. So the thing is, even though you can't see it with your eyes, if at night you set up a little tripod in your garden and you put a little camera, for example my own digital SLR camera I had, so I put that on a tripod, and we were all part of this little project where we even encouraged students to be part of it. तो रात के time moon की तरफ आपने बस अपना camera focus करके रखना है, and at a regular interval of let's say 2 minutes or 3 minutes or 5 minutes, the camera should click a picture. And you focus it on the moon, you cannot see anything. You can just see the moon, of course, as you see with your naked eye. But when you focus the camera onto the moon, and you can do it yourselves at home, and then you look at your pictures, I'm sorry, can I copy both of the talks so that there are 2 of them? You've copied them, okay. So I'll just finish this little story. And when you look at these pictures later on the computer, you will find birds migrating across the moon's surface, because the moon is reflecting the birds across them, and you can never see those birds in reality. But on the camera focuses and clicks them and captures the birds. So we record how many birds went each time, and there are groups of these flocks of birds. They are all dark, looking like crows actually, but they are big, large-sized birds. And so data was collected by different students and researchers all over the country. We kept it open for everybody to participate. And then the data was collated in Bangalore, where this project was based at the Indian Institute of Science. And the results were astounding. One researcher, one human being in one city could not have collected information on bird migrations the way it was done by all open people from different fields, not even from science, people from outside of science also, anybody who had wanted to participate in the project. So coming back to my talk, that's all I had wanted to let you know. Even smaller equipment can sometimes let you see astounding events that are happening in the sky, in space, where we can't actually see them. So daytime migrations are not possible because you can't see those birds. But nighttime across the moon, you will have these pictures of these little blobs or large-sized blobs which can allow you to capture bird migrations. So fine. I'll get to my talk now. So as Dr. Avasti has said, I work in the area of agriculturally useful biotechnology. So genomics is the specialization that I and my group, my laboratory, we try to carry out at Delhi. So I'm from New Delhi, and from the National Institute of Plant Genomic Research. So since you're all school children, I possibly will have to explain to you about genomics and some of the very basics of DNA, which I have not put in the slide because I was thinking that this would be a group of college-going students, postgraduates, and school students. So now there are school children, and all I can say is that we've all got DNA in our cells, and the DNA is the code of life. And to decode information from DNA, from our cells, or from any organism, what we do, a very recent technology, is next generation sequencing. Basically, you take out the entire DNA from one cell of an organism. That could be a single cheek cell, or it could be a little cell in the leaf of a plant or the root of a plant. And then you extract out the DNA, and then you hack at it and you try to break it chemically into very tiny pieces or units called bases, or strings of bases. Quite often you can't break DNA completely into tiny bases because DNA is a very long string. So what you can do is break it into tiny strings of bases. And then the task is to assemble all of those strings, because now you've got small pieces that you can read. And there are only 4 letters to the language of DNA, A, C, T, and G. So these are 4 nucleotides, adenine, cytosine, thymine, and guanine. Okay, so these 4 and their combination, so we get these strings and then we stitch together the strings into a long sequence where we try to reproduce every chromosome in your DNA or the DNA of a plant. In my case, we are sequencing the DNA of crop plants, which are important to Indian farmers. So very recently, we have decoded the entire genome or the DNA of the chickpea, the desi version of the chickpea, which you might have heard as the chana or chole, jo aap khate hain. So that is the plant which is probably one of the most important legumes across the world, and certainly very important for India. After grains, legumes are the second most important crop anyway. So we have decoded the chickpea genome at NIPGR very recently, and we are carrying out now. So after hacking the genome into pieces, then stitching it back together, and then getting the entire sequence, assembling it, we have to analyze it for where are the genes, because the genome contains genes, and the genes contain information that is going to be making all sorts of molecules, biomolecules like proteins or ribosomes or tRNAs or transposomes, etc., etc. So then we study that genome sequence and we try to analyze the information that is there and extract information that might be useful. So if I'm an agricultural scientist, I will be specifically looking for information that will help farmers improve their crops for nutritional benefits, for nutritional efficacy, or especially in India, droughts can be a very serious threat. So I will be looking at a genome sequence specifically in terms of trying to see if there is any gene that might allow tolerance to drought or stress tolerance of a different kind, whatever is hampering that specific crop for our farmers in the fields. So the idea that I'm going to give you today is about different kinds of plant genomic advances that we carry out in research in order to be able to help farmers in agriculture in the country and across the world. If you have any questions which you think you're not able to understand, please ask me, because I understand that you're school children and what I might say might be a little fly away like a bird migration over your head. So in case you want to stop me and ask me at any point of time, please do that and feel free to do that. Okay, so genomics in sustainable agriculture. We all know that the declining water resources and increasing climate instability are threatening the productivity of plants on a worldwide level. And there is a demand for healthier, safer, sufficient food supply, which is constantly growing. And we know that there have been numbers of gains in production through plant breeding. The traditional method of improving a plant used to be plant breeding, like hundreds of years ago. You know Mendel's experiments probably for genetics, and you know that he was breeding pea plants. So that was the mechanism at that time. It is still a very important mechanism for improving plant varieties, but the gains that we achieve through such methods are currently not able to offset the losses from the kinds of stresses that these plants and these crops are suffering, such as biotic stresses and abiotic stresses, internal and external stresses, etc. So responses to such stresses are complex multi-locus traits which make it extremely difficult to improve the quality or the yield of a crop through conventional methods or traditional methods of breeding. Therefore, it is extremely important now to use targeted specific research using genomics, since it can play extremely important or pivotal roles in maintaining and improving the yield, productivity and sustainability of crops. So now researchers are beginning to discover how plant genomics can play a significant role in enhancing the quality and quantity of crops. Plant genome research is needed as the foundation of an entirely new level of efficiency and success in the applications of genetics and breeding of crop plants. So knowledge of the plant genome or the DNA of plants could also serve to reveal genetic vulnerabilities in plants. Currently, we call as weeds. Some of the most important weeds are plants which are closely related to crops themselves. So it's extremely important to understand the weeds as well as the crop plants. Not just that, it is also important equally to understand the genome of the pests that are infecting the plants. So as an agricultural scientist, for me it is equally important to know the genome of the chickpea as the genome information of the pathogen of chickpea or the weeds that are growing around chickpea. Alright, so proteomics, genomics and bioinformatics are emerging as important areas of research in this field, in the field of agriculture. So there is a huge potential for plant genomics. We can improve quality, safety and diversity of food and other products of plants. We can have greater food security assurance worldwide in the face of a doubling of world population and declining agricultural and quality land and quality water for irrigation. We can have a cleaner, healthier environment, etc. We can use an expanded variety of plant products, and because of the expanded knowledge, further reduction in any potential environmental or human health risk that might arise with crop plants as a consequence of genetic modifications. So genomics is a field of research that relies on very high throughput technologies. These high throughput technologies could be of two kinds. Hello. So one is the experimental kind, the other is the non-experimental, I shouldn't say non-experimental, but computational kind. So there is the in vivo or in vitro research, and there is on the other hand the in silico research or computational research, which we call informatics, bioinformatics. So in case of experimental research, for genomics, we require very high throughput automated sequencers. So these are the machines that are going to hack DNA into small pieces and then return pieces or strings or units back to you. There are going to be robotics, microarray spotters, high throughput genetic techniques, etc. And in the computational side of it, there is going to be the sequencing, which is the assembly. So you first carry out sequencing, you get the pieces, then you join them together. So you assemble them together into chromosomes or pseudomolecules or linkage groups. So this process is called sequence assembly. And then comes the job of annotating the sequence for where are the genes, where are the exons, where are the introns, where are the repetitive sequences, where are the regions that are junk DNA. So those kinds of things. And then comparative genomics. Once you have a genome sequence, once you've done some kind of an analysis, for example, the human genome has about 30,000 genes, the Arabidopsis genome has about 25,000 genes, the chickpea genome has about 27,000 genes. So now that we know that so many genes are there in the genome, comparative genomics is the next thing to do. You look at 2 or 3 genomes and try to compare them. Are the same genes present across all genomes? What are the differences? Which genes are unique to one genome? Which genes are only present in animals? Which genes are only present in plants? Or which genes are only present in bacteria? Or which is the set of genes which is present only in a particular kind of plant? Let's say the land plant, etc. So then you get such, the field that is related to doing this comparison is called comparative genomics. And then we have network biology, which is becoming systems biology, I might call it, which is becoming extremely important in making intelligent inferences, because the kind of data that we are getting from genome sequencing projects is enormous. It is huge. Nobody has an idea how to look at all of this information, millions and billions and zillions of strings of nucleotides, and how to extract information that will be useful for mankind. How to extract information that will improve our own knowledge of nature, of how things work around us, what is there inside of us, outside of us. So systems biology is another way of looking at it, and cloud computing is becoming extremely important. Sitting in Jabalpur in your little school or a college, you probably do not have access to supercomputers But some of you might be having the curiosity in your mind to be having a question that you would like to answer, to be wanting to do something which is over and above the infrastructural facility available to you. Therefore, that is where comes in cloud computing. So all of you, I don't know if you're aware, for example, can log in remotely. If you have the internet connection, you can log in remotely to the mighty supercomputer available at the IIT Delhi, at the supercomputing facility at IIT Delhi. For free, you can get a login. For students, it is completely free. So you are not limited or restricted by the kind of infrastructure available to you. At home, probably you would have laptops or smaller PCs. Even in school and college, you would have good machines, probably workstations, but you can't use them at the same, at the kind of, in case you want, in the way that you probably want to use them. And even if you get access, you may not have the programs that are required for doing these analyses. So the supercomputing facility, the SCF at IIT Delhi, has an online registration form. You just put your email ID, your name, and you tell where you're from, and they will give you free access to one of the best supercomputers in the country, and you will have a login ID and you will be given space and you can do any kind of computing you want, and there's so much freely available data. You are welcome to join into research and ask a question and address a biological question of interest to you. For example, as I said, you are most welcome to be part of the bird migration project. In the same way, you are welcome to participate in genomics or in any other kinds of science that probably requires computing prowess. So cloud computing has made it possible that we are not anymore limited by the computational infrastructure that we have or do not have. This is a research outline for plant genome research, and sadly probably you can't see it so clearly, but there are, in case it helps, there are 3 or 4 major levels shown in different colors over here. So first you need germplasm data. You need plant data to carry out plant genome research, which can be got available in terms of cultivars or varieties of plants and natural populations or gen bank material, etc. So these are stocks, generic stocks. Then you get maps out of them. So you get express sequence tags, you get physical and genetic maps, and you start sequencing. You get the DNA and you get the sequence itself. After getting the sequence, the idea is to do analysis. Analysis can be of different kinds, as I said: comparative analysis, single standalone analysis, stress responsive analysis, I don't know what all. So functional analysis or QTL based analysis. So I'm not going to go into details of any of these, but you're most welcome to ask me specific questions if you want to later on. And then comes the level of, finally, the hope is to get candidate genes for important agronomic traits. For example, an important agronomic trait of chickpea, of any legume plant, is to be able to capture nitrogen, what we call as nitrogen fixation. Nitrogen fixation is not going to be possible by any other kind of plant. Only legumes can do it because they have this specific guy called leg hemoglobin with them, and they have some microbes that are helping them. But they are able to have an interaction between the plant and microbe through certain specific genes which make certain specific proteins which enable the fixation of nitrogen. And a lot of that depends on a certain metal. Very recently, last month in Science, it was published that an iron-like complex is involved in nitrogen fixation. Now that we know that, it is possible to put that iron-like complex into any other plant which is unable to fix nitrogen from the atmosphere, and therefore that would be a great leap for mankind, for agriculture, for biology, to be able to enable nitrogen fixation by plants that are not legumes. So these are kinds of things we want to do. We want to have candidate genes or proteins that are going to be important for critical agronomic traits. Okay. So there are lots of large-scale sequencing projects that have been going on in the world. One of them is the earliest was the Arabidopsis thaliana. In 2001, the first plant was sequenced, and over the years, over the past couple of years, past decade, lots of other plants have been sequenced. As I told, at our institute at NIPGR, we have been part of the group that has sequenced the chickpea genome. So there are lots of different genomes, and here is a picture of the tomato genome article that was published in Nature last to last year. So currently, very interestingly, unlike plants, unlike animals, there are more than 40 or 50 different plant species for which sequencing is underway at various levels of completion. So that's one thing. And therefore it makes plants a very interesting field of study because you have so much data available. For animals, there are probably 4, at best 5 animals for which the genome sequence is available. But for plants, the data is increasing at a very large speed, at a very, very fast rate. So we have a lot of data for plants, and we have plants which are not just, we are not limited anymore to model plants like the Arabidopsis. We now have plants which are genomes from trees, we have genomes from smaller plants, the older plants like magnolids, we have genomes from dicots, we have genomes from monocots, we have genomes from mosses, bryophytes, pteridophytes, we have genomes even from fungi which are actually at the lining between plants and animals, but we have these genomes available so we can compare the genomes to see what kind of sequences are specific to to different life forms. Alright, so the thrust areas in plant genomics for agriculture are listed here. One of them, of course, is plant pathogen interactions and plant insect interactions, because insect and pathogens are extremely big damage causing organisms for plants. So determining the evolutionary history of plants using sequence data from conserved genes, it's extremely important to understand how plants have evolved. Evolution of plants allows us to carry out or plan future experiments also. Let's say if you want to carry out a breeding experiment and it is known to you that plant A is evolutionarily closest related to plant B, then you would like to, you know already that there are common genes between them, a large number of those. So you would like to carry out, probably you would like to breed with another plant which is a little distant so that you get new genes within yourself in order to improve your hybrid vigor. So evolutionary distance and evolutionary history can be very helpful for agricultural purposes. Light perception to set circadian rhythms and determination of developmental patterns in plants. This is one specific area where I am working in my lab, and later today I will be presenting the work we have done in case of developmental patterns, especially floral transition in plants, and the way we have used systems biology to answer to find answers to questions that were not addressable earlier, before the genomics era began. So increasing nutritional values of crops, of course, that should be extremely important. So at NIPGR, there is a group of senior scientists who have developed a tomato variety which has a huge shelf life. We are also trying to improve the potato in terms of various factors or features. So yes, best genomics indeed. So for example, the spider mite is an extremely dangerous creature for certain plants in the northern Indian region and across the world also. So the genome of the spider mite is now available, and there was a big question about the spider mite earlier, and it was not understood how it does this. It is able to change its own genome in response to the plants. Quite often what happens is, perpetually in nature, plants and their pathogens or their enemies are fighting with each other, a very interesting war which is perpetually going on. So plants are able to alter or make new genes in response to a new kind of threat, and they are able to handle or handle themselves or sort of survive or tolerate the enemy or the threat by way of these new genes that they can, which are called resistance genes or virulence genes, etc., etc. So this guy, the spider mite, was uniquely threatening because it could easily overcome, just like our antibiotics are there, the microbes are overcoming antibiotics by resistance very soon. The spider mite could do the same. Only the genome, when it turned out and revealed the secret of the mechanism by which the spider mite is able to overcome the plant's constant efforts of resisting it, was very interesting, and I will come to that later on. So best genomics is equally important for agricultural genomics, and there is the role and enormous success of the RNAi technology, which I think as school children I should not have put that slide at all, probably for senior students later. So then the question arises, in what way are we going to get to the road of commercialization? To what extent do our research or the technology generated will actually make a difference to mankind, and how long can it take? It can take a large number of years. It may never happen. It may sometimes happen. So everybody's probably heard of BT cotton, but this is an example of the golden rice. So I don't know how many of you have heard about golden rice, but it is the result of an effort to develop rice varieties that produce provitamin A or beta carotene, since the traditional rice varieties do not produce this specific vitamin. So the two genes were taken from another plant, daffodil, and one was taken from a bacterium, and these are the guys that can make that vitamin. So these genes were inserted into the rice genome so that the rice grains could also make that vitamin. And these three genes together produce a series of enzymes which are necessary to convert the substrate into provitamin A. Okay, so golden rice contains beta carotene, and this method of ingesting golden rice or using golden rice in your food could be one other way to address the vitamin A deficiency which is rampant across the world, especially in children. And very recently, in 2012, there was some research that was published in the American Journal of Clinical Nutrition which showed that the beta carotene produced by golden rice is as good as the beta carotene provided in regular oil for children. So there is no harm in being able to take. Of course, it will take a long time probably for India to even consider, or many countries to consider golden rice, but this study is a step towards a future where we might actually be able to give children the golden rice. And the study demonstrates that children who are among the most vulnerable to vitamin A deficiency could indeed benefit from this form of rice. So BT cotton, of course, coming back to it, which was the original one, cotton is the most important commercial crop of India, often referred to as white gold also. And the most important insect pests that are affecting cotton are different kinds of bollworms, aphids and thrips, etc., and leaf-eating caterpillars. Of these, the American bollworm alone can cause a lot of yield reduction across the world. So BT cotton is an insect-resistant transgenic crop. In case, for those of you who are not aware specifically, it can overcome or combat the bollworm because it has a specific gene that is toxic to the bollworm. So that gene was transferred, and it was transferred to cotton from a specific soil bacterium which had that toxin. Most of the toxins in nature are present in microbes. So we used the microbes to get those toxins, but this was relatively easy because a single gene had to be transferred. Unlike the golden rice, where you don't have a single gene or a single enzyme, you have a series of enzymes carrying out a pathway, and vitamin A, provitamin A is not going to be formed unless you have each member of that, every single enzyme and the gene there. So putting one gene is not such a big problem as putting a series of genes and proteins into a new organism. So that was much tougher, but it has been achieved, giving more hope to agricultural biotechnology. So coming back to BT cotton, the first time BT cotton was introduced or economically grown was in the United States in 1996. In 1995, the government of India had permitted a certain small import of some of these seeds, and to try and see if we could enable it in India. And Monsanto Myco tie-up, which probably some of you have heard of also, was able to show trials that it can work in India also. And in 1999, the trials were presented across 40 locations. Finally, in February 2002, the ICAR, which is the Indian Council of Agricultural Research, the senior most authority for presenting reports to the government at all, submitted a positive report to the Ministry of Environment on the field trials of BT cotton. And last couple of years back, we celebrated a decade of successful cultivation of this crop, which has achieved enormous and phenomenal success in transforming the cotton crop into the most productive and profitable crop in the country. Alright, very recently, as I told you, the maize genome was sequenced, the tomato genome was also sequenced, the chickpea is ours. So what kind of information can we get from these genomes? This is the corn genome. The corn genome has about 17, I think, chromosomes, 17 or 12 chromosomes here. So what you are seeing is a comparison of the corn genome with the then available rice genome and the sorghum genome also. So every circle represents one kind of information, and in this form it is much easier to see who is similar to whom. So computational biology is becoming more and more important in agriculture and in all forms of life science. With computational biology, it is possible to visualize the entire genome of an organism, not just one organism but multiple organisms, in a superimposed manner, so that you can compare the things that are comparable. You can find out where are the common stretches. You can find out where are the unique differences. Earlier you had to take the millions of long stretches of DNA and blast each one with the other genomes and try to see where are the highest matches available. Now you can just put it together using such programs. So the maize genome sequence includes locations on chromosomes of interesting repeated regions of DNA called the centromeres that are responsible for faithful inheritance of these chromosomes by daughter cells. And the maize genome sequence represents a major watershed in genetics, agricultural genetics, because it promised to advance basic research of grains and help researchers, scientists and breeders. Resulting improved strains of maize can, for example, produce larger yields, show resistance to disease, and offer efficiencies in nitrogen use that would enable farmers to reduce applications of costly, polluting fertilizers and tolerate changes in rainfall or temperature accompanying climate change. So the genetically modified corn is a recombinant corn plant with agronomically diverse, desirable traits. Alright, I'll try to rush through the talk now, but I have to tell you about the spider mites. So a Canadian research team very recently, featuring scientists from a large number of countries. So again, collaborative research is becoming extremely important in future. I don't think there can be single units of researchers collaborating not just with your own specialized area colleagues, but with colleagues from other specializations can be equally important. So these were a couple of guys who got together from Spain, Belgium, France, Portugal, USA, Chile, Germany, Switzerland and Canada, and unveiled the first genome of the second largest group of animals on the planet, the chelicerates, by sequencing the genome of the spider mite. This guy, the spider mite, is one of the world's most cosmopolitan pests. Why is it cosmopolitan? It's a page 3 pest, because this guy can change itself and they are destroying a huge number of potential crops, but its specialization is that it can develop resistance to pesticides very fast. Within 2 years of the introduction, spider mites are able to overcome any new pesticides that you decide to put on the field. So the scientific team uncovered the genetic basis for the mite's ability to feed on so many different plants, discovering that what this guy can do, the spider mite is able to multiply and evolve new genes at a very fast pace in order to detoxify the toxic plant molecules, and very surprisingly, it also hijacks the detoxification genes from bacteria, fungi, even other plants in order to combat plant defenses before incorporating it into its own genome. I don't know whether some of you can even understand the huge gravity of this discovery. An insect, an animal, is able to hijack detoxification genes. So it recognizes what are the genes in the plant or the bacteria. Bacteria are the guys who have most of the toxic genes. So what are the genes that are going to be toxic to me? And therefore, make a copy of those genes, get that copy, make proteins, and try to evolve my own genome in such a way as to overcome the toxicity of those toxic genes. And in addition, take that stretch and incorporate it into my own genome, a hugely incredible kind of biotechnology that the mite is carrying out within itself. So it incorporates its own genome so that in future it may not forget that the toxicity had happened, let's say a number of years ago, using this copy of the gene. Every time it is able to evolve in this manner. And this would never have occurred to anybody working on this pest unless and until the genome was out. When the genome happened and the genome sequence came, and that circular image I had shown you for the crops, the same kind of work was done. Usually when a pest genome comes out, what do? We take the pest and try to compare the genome with other pests, because you like to compare people who look like each other. That what are the differences between this pest and another pest? But what the researchers, what occurred to them was, and it's an incredible paper, what occurred to them was, don't compare the pest with other pests to see why it is better than other pests. They compared the pest genome with the plant, with the host genome. So when you compare the host and the pest, there was no reason to expect any similarity between an animal and a plant in important places, except for the constitutive areas, of course. But they found huge patches which were common. So in the previous slide, did you see these arcs, the lines which are connecting different patches together? These lines mean similarities. So the chromosome number 13 of rice has a lot of black lines connecting it to the chromosome number 9 of sorghum, which has some black lines connecting it to a large number of genes in the chromosome number 1 of corn. This allows you to see that there are common pieces or patches or regions between different plant genomes, and it is expected. Plants are highly polyploid and they have been evolving in such a way as to have duplications, incredible numbers of duplications, and that is how they have evolved. But the mite has evolved in a similar manner. An animal has evolved in such a way as to take pieces of the plant genome. So when the plant genome, the host plant genome, was compared with the spider mite plant genome in a similar picture, they found a large number of specific chunks of black lines going from one specific place on the mite genome to specific areas on the plant genome, on the host genome, and that is how they discovered that this guy is hijacking the DNA of the plant. So that's an interesting paper. Anyway, so coming back to pest genomics, there are lots of things that people do in pest genomics. Disease and pest identification is important. In case of working it out about handling pestilence, you try to spray the landscape, have fungal controls or other kinds of disease controls. So all of these are part of agricultural research subjects in pest genomics area. RNAi, I'll skip it today. So genomics in tree breeding is also there, and there are lots of ongoing genomics efforts for sustainable agriculture in India and across the world, including researchers at Cornell who are currently working to enhance the specialty crops by training hop growers, promoting disease-tested grape wine, and developing weed suppressors. At UC San Diego, there are certain biologists who have succeeded in genetically engineering alga or mosses to produce a complex and expensive human therapeutic drug to treat cancer. Researchers examining how the hormone jasmonate, a plant hormone, works to protect plants and promote plant growth have revealed that a transcriptional repressor of the signaling pathway for that hormone makes its way into the nucleus of the plant cell. Identification of genes and alleles responsible for natural genetic variation in crops, etc., etc. There is also some very interesting work going on in the sea buckthorn. And at NIPGR, where I come from, this is a satellite image of our institute, and these are our laboratory areas, these are our fields all around, and this is where we stay so that we can have direct access to the laboratory and its infrastructure 24/7. So we have a lot of initiatives going on in genomics. We have, as I said, sequenced the chickpea genome very recently, and genomics can be effective in enhancing the yield of tropical food crops like other legumes as well. There is a lot of work going on in nutritional genomics. I told you we have got improved genes and enzymes which can help to improve tomato and potato, and there are pest genomics efforts going on. We have not been part of any pest genome sequencing project yet, but there is a lot of work going on in the plant-pest pathogen interaction research area. There is lots of stress genomics, abiotic as well as biotic, going on at NIPGR. We are involved in building databases for genome diversity and online servers and programs for which my laboratory is also participating. We hold annual trainings and workshops. Dr. Ravasti told you at the beginning that I could not attend this life science meeting of JIGYASA last year because we were conducting a workshop. So every year we conduct this workshop at NIPGR where we inform students, or we try to carry out demonstration sessions, and this workshop. These workshops are free for students. In case any of you or somebody you know are interested, please ask them to write to us, and at the next workshop you would be most welcome, of course. So we have annual training programs also, where we train students for 2 months to 6 months for different kinds of work, research that we carry out. So the message I would like to leave researchers or potential future researchers with is that whenever you're thinking about a new project, or whenever you're curious to answer a specific question, at least in case of agricultural genomics, please make an effort to listen to the farmers and the real users of your whatever question you're trying to ask or address, and then design your project based on the requirements of the industry in order to make the research more applicable, which is extremely important for people like us, because we are in a national institute where developing technology is one of our major mandates, and in order to be useful, we have to be able to keep in mind what the farmers require. And with that, I would like to close my talk, and thank you all for listening to me.